Okay, so it's a great pleasure to announce the winner of our big prize, which is the Hooke Medal. And it's particularly wonderful for me because I was awarded the first Hooke Medal when I was, 10. And this year, the prize winner is McAinsh. Now, I want to say a little bit about the Hooke Medal. So the reason it's called Hooke is because Hooke was the first person to see cells down the microscope, so that's very appropriate. And the second thing is that now, not when I won it, now it's awarded to someone who's within 14 years of starting their own group. So it's kind of a mid-career award. And there was very steep competition this year, and so it's absolute pleasure to have Andrew, who's done amazing work in the field of cell biology, to win this prize. So Andrew, come up here.
Thank you very much. So first thing, of course, I have to say, I'm very honoured to have this, and I have to say I never imagined I'd actually be standing in this place doing this. And actually, when I got asked to give this talk, Anne said I should talk a little bit about how I ended up here, right? So I thought I'd maybe do a little bit of that, talk about some science as well, and try and sort of bring things together a bit at the end about how we really see the future of cell biology and what we think are the big questions for the future, and try and do that. But it is important to thank British Society of Cell Biology, and I think also really important to say that actually all my work, it really depends on all of you as well, because in fact it only works in the context of a great cell biology community and other people doing fantastic work around it, and it's based on those discoveries that we're able to do some cool experiments as well. So I think the whole community is really important. And related to that is the fact that, I obviously wouldn't have this without all the brilliant people who have actually worked in my lab, because actually they have done the majority of the hard work over the last few years. They've worked super hard. Some of them are here today. Sarah's at the front here today, and my students from my lab now are here as well. And I think that, I've got to say a big thank you to them. It's something for all of us to share, not just for me. Now, today's talk, as you can see here, is going to be about chromosome dance steps. I think that's apt because I think that the way mitosis works, and we've seen some talk from mitosis this morning, is that you actually do need to do this really beautifully choreographed movements in order to complete mitosis error-free, and that's very important. I've put up here a few dance steps. There will be a prize later for anybody who can tell me which dance steps are on the screen. These are not mitotic dance steps. We'll talk about those in a minute, but so there may be a prize later for that. So let's talk about the chromosome dance steps. But I think before talking about them in detail, I think it's worth talking about some context for those movements, because they don't happen in isolation. And I want to show you this first movie first. I think it's actually my favourite movie, and it's not mine, right? Okay, this movie is from Geraldine Hartshaw, and she is a collaborator of mine in the University Hospital. And this is from the IVF clinic. So what you're actually seeing here, this is a single human zygote, okay? And you're going to watch the first cell divisions of a human being be created. And I think that, we've seen loads of cool cell biology, mechanistic cell biology, but I think it's always worth thinking about the fact that actually this is actually what I think it's really all about, is actually how do you build this human being in the end, starting from one cell. And you have to go through, as you can see here, you start with one and we have to end up with something like 40 trillion cells. And how that works, I think, is amazing. And I think that to understand these initial divisions is going to be very important. And this is my first slide, and my last slide is going to come back to this exact question, okay? But you've got to hold on for that. But we're going to talk about that right at the end. So that's 2.5 days, okay? That's the first initial cell divisions. And obviously, as I'm saying, those cell divisions are important because you need to make sure that in each of those divisions, you accurately segregate your chromosomes, okay? You need to make sure you have the right number every time you divide. That's essential, okay? Now, why is that important? It's very obvious why it's important. Here is the karyotype from a normal diploid human cell. You can see it's 46 chromosomes there. And we all know, we're all very familiar with the concept that when this goes wrong, this causes problems. So here's an example of a karyotype from some cancer cells, and we know that chromosome instability, the development of aneuploidy, you heard Susanna talk a bit about aneuploidy earlier this morning, is really important with respect to cancer development, okay? So it's important that the cells maintain this. In the context of that human oocyte that you saw there, you also saw Binyam tell you this morning about obviously the importance of those initial meiotic divisions that create that single cell at the beginning are important because if it goes wrong, you end up with these developmental syndromes. I think that's a really important human problem as well, and both these things make it a really interesting and important area to try to understand. So how does it work? How do you actually segregate your chromosomes accurately? So I'm going to start with this cartoon here. This is like the simplified version of everything, right? So in red here are the microtubules, the mitotic spindle, okay? The two blue things these are the two sister chromatids. And you can see these two sister chromatids here are connected together by these little white discs. These are the cohesin molecules that are essentially acting as the molecular glue that's going to hold those two sister chromatids together, okay, until they need to be separated. But Biniam kind of mentioned it earlier, but I think the most interesting part, of course, is these green dots here, okay? These green dots are the kinetochores, okay? And it's these kinetochores that we've been working on for a long time now and are really trying to understand. And these kinetochores, I think, are at the heart of how this process works. This process could not work without these kinetochores. Okay? So here's a cartoon of what the kinetochores can do. They're pretty amazing. They can slide along microtubules, as you can see here. They're able to grab and grip microtubule plus ends. They're able to coordinate their movements to actually move the chromosomes into the metaphase, the middle position of the cell. And then at the exact moment, they can tell that they're correctly attached so that they can dissolve the cohesin and drive the segregation of chromosomes in anaphase, okay? And you can see along the top here, these are the key processes that I think we need to understand. So there's this sliding, there's the ability to catch microtubules. They're able to hold on to those microtubules somehow, okay, under pulling and pushing forces, okay? And then eventually they need to coordinate this such that they know when all the chromosomes are correctly attached to segregate the chromosomes. And so it's those processes that we are trying to understand. So here's what I think is a good cartoon of a human chromosome, because what I've shown you before is a simplified version. The interesting thing about the human kinetochore, which are these red, you can see the kinetochores here, these are these red things here. You can see they attach to these bundles of microtubules, okay? About 20 to 25 microtubules in a human cell. And somehow these bundles of microtubules coordinate their pushing and pulling movements to drive oscillatory motion of the chromosome. And then, as I've just told you before, eventually lead to the dissolution of the two chromatids and the separation of the chromosomes like that. And so it's that process, but by which, how do those kinetochores coordinate all of those microtubules that they're bound to coordinate those processes? Essentially, kinetochores are like Pac-Man, right? So effectively what they do is they effectively chew their way along microtubules and maintain that attachment. And that's actually the fundamental mechanism that we need to understand. We're going to talk about the molecular details of that later. And actually, the term Pac-Man was the original term coined to describe the motion of the chromosomes towards the pole. They essentially are operating like Pac-Man. Now, there's other systems in place here that ensure this works, okay? The first one is this. So if the two kinetochores, as you see them here, are attached correctly, they generate pulling forces, and they actually effectively generate tension across the chromatin that links the two sisters together. And the kinetochores know that they're under tension, and when they're under tension, they strengthen their attachments, okay? So if that's important, because if they're misattached, if they're not correctly attached, then they lose tension, and then they will effectively let go of microtubules, and they will try to start again. So you have essentially an error correction process that's running to try and ensure that all the chromosomes are in this biorientated state. That makes sense because that's the only geometry that's actually compatible with accurate chromosome segregation. No other geometry would actually make sense, as far as we know. So that's one mechanism. The second mechanism is highlighted here. So this is just an immunofluorescence image of a mitotic spindle. You can see actually most of the kinetochores there are aligned down the spindle equator. They're colored in red there in this particular image. Microtubules, of course, are blue in this particular image. You can see something's gone slightly wrong here, okay? So this chromosome that's attached down here, you can see its kinetochore attached to some microtubules. It's clearly improperly attached. And because it's improperly attached, it binds this particular protein here called MAD2, shown in green. MAD2 is a spindle checkpoint protein. MAD2 binds here and generates a wait anaphase signal. This whole cell now will wait. It will not enter anaphase until that problem is resolved. This is an exquisitely sensitive system, and it prevents the loss of single chromosomes. Okay? It essentially monitors attachments and ensures that everything goes well. And we're going to talk about that process later on as well. So to summarize that first kind of introduction to the important concepts in kinetochore biology, we need to understand how they attach to microtubules. We need to understand how they grip microtubules and remain attached to them under pulling forces. We've got to understand how they're able to sense tension or sense other events and change their behavior in accordance. And we have to understand how they're able to generate this soluble weight anaphase signal that enables the process to be error-free. So here I think are the big questions. So what are the components of a kinetochore? I haven't told you anything about that yet. What are they built from? How do they assemble? How do they form during the cell cycle? How do kinetochores actually generate force? What are the molecules required for that? And which molecules are required for the sensing of forces? And how do they then respond to those forces? And those are the big questions. And that's what we're going to talk about. But before we talk about it, I want to tell you how I ended up working on kinetochores, because I think that's important. And it actually started here in Manchester. I was an undergraduate in Manchester. I did my first degree here. I actually grew up here as well, which makes it even more kind of fortuitous, or the stars kind of aligned. I like to say the kinetochores kind of aligned, actually, in a sense, right? Because I'm able to come here to have this today and talk about kinetochores to you in what is my kind of my hometown, right? Which is fantastic. So I'm going to embarrass Ian, who I know is here, because Ian played a really important role. Ian lectured me on the cell cycle when I was an undergraduate. I went to Ian's lectures. He had great lectures. He talked about actual science, papers. My other student friends weren't so always keen, but fantastic, and made me think, I want to work in that field. Science is exciting. We talked about some of the latest discoveries that had been made. Ian at that time had just come back from Japan and was full of all the exciting things that are happening in the cell cycle world. And I thought, that's what I want to do. I want to work on that. Ian actually didn't encourage me to go to his lab. He actually encouraged me to go to different places and learn new things and see new things. That was great advice, really important for everybody, I think, like to spread out and go and see new things and learn new things. And he told me I should go to this new guy in Cambridge, Steve Jackson, who was just getting going, doing his lab there, working on DNA damage, DNA repair. So I took that advice and I went to Cambridge to do my PhD, where I worked on budding yeast DNA repair. I have to say, working on DNA repair was great. I had a great time, but John Pine's lab was next to me. John Pines at that time had started doing live cell imaging in human cells. He was injecting fluorescent cyclins and things into things and doing live imaging. And I was watching that and thinking, that is really the right thing. I like this live cell imaging thing. The ability to actually understand these processes and the dynamics of these processes, I think is really important. I thought I'd like to do that as a postdoc. So to do my postdoc, that's when I got involved in kinetochores, because I went to MIT. I joined Peter Sorger's lab, MIT, to work on kinetochores. I stayed working on budding yeast, but the idea was to now do live imaging and look at chromosome segregation, in particular kinetochores, and try and understand how they work. So here's my first photo. This is me at the dinner table at the end there. That's Peter at the front there, and some of my colleagues in the lab at the time. And we had a fantastic time there in Boston, working on kinetochores. And this is actually what the kinetochore was like in the textbook and the papers when I went to Boston. It looked like this. It was really quite unexciting. It literally was black and white, right? It had like 8 components, okay? This was a budding yeast. All the work was done on budding yeast at that time, because budding yeast kinetochores only bind one microtubule. Unlike those complicated human things, budding yeast bind one microtubule. So there was only 8 components actually known. And so the idea was to sort of study these kinetochores. And at that time, people started doing a lot more sort of proteomics, mass spec, analyzing new proteins. And we thought, actually, maybe we should try and have a look and see whether there are other kinetochore components. How complicated is this structure? Or is it that you literally just need one kinesin motor protein and that's it? So that's what we did. I'm going to show you, the question was that, because we know that even something as simple as a car is created from huge numbers of parts. And it seemed that to understand how the kinetochore worked, we needed to know the parts list. That seemed to be an important first step. And so that's what we did. We did a whole series of affinity purification and mass spec, cutting bands out of gels, doing mass spec, sequencing these proteins, labeling them with GFP, showing they went to the kinetochore, and trying to basically map out the components of the budding yeast kinetochore. Not just me, I have to say, as I said at the beginning, this was me and many other labs in the field. I remember going to talk about this work at Cold Spring Harbor at the yeast cell biology meeting, and there was 3 talks. I was the third one, and the first 2 were on exactly the same thing, right? I think that was kind of worrying at the time, but actually in retrospect, that was a good thing. Everyone was kind of putting this thing together. It was an exciting field at the time. We were learning new things about how the kinetochore worked, and we were building up this map of all these proteins. And then eventually we did all of that, and we came up with this model shown here. This is from a review that we wrote. And the basic concept was at the time was, in this review, was that the kinetochore actually is not like a ribosome, right? It's not one big solid protein structure, okay? It's actually built from lots of these different subcomplexes, which are sort of highlighted here. The idea is that these subcomplexes dynamically come together to form a kinetochore. And this is important because when you break open the cell, the kinetochore just disassociates into many subcomplexes. And so that was an important concept. And so we've kind of imagined actually that kinetochores are a bit more like, say, a transcriptional enhancer or something where proteins would come and bind, or RNA polymerase or something like that, and not so much like a single ribosomal complex. I was very proud of this picture here on the left here. It took me a long time to draw this. My wife made this for me though, which I think is the best thing ever. So she made me a kinetochore mug, you can see that. I'm sure if you ask her nicely, she's here today at the front, you can ask her nicely, she may paint you a picture of your favourite molecular structure too. You have to come and ask her afterwards, okay? And this is sitting on my desk to this day. I really like this. This is great. So this was the model on the left here that we also drew, which was the idea that a budding yeast kinetochore binds a single microtubule, okay, like a socket, okay? And there would be this sort of like ring complex, this green ring that goes around the top. That ring complex was also identified, that was the DAM1 complex. Not by us, but again, a really important concept. This on the left here is an electron microscopy picture of a purified budding yeast kinetochore from Sue Biggins' lab. This is one of my favorite papers, actually. You can see the ring around the microtubule, and you can see the kinetochore really is pretty much operating a bit like I've sort of drawn it there in a sort of thought on the left, right? It seems to look like a socket, so the microtubule is embedded and engaged into this multi-protein structure. That seems to be how it works. Now, having identified all of these budding yeast proteins, we thought maybe it'd be interesting to find the human versions of all those budding yeast proteins that I've told you about. And so now I want to introduce my really good friend and colleague here, Patrick Maraldi. And Patrick and I continue to collaborate to this day. But Patrick and I were both postdocs in Peter's lab at the time, and Patrick came to work, he came from Eric Nigg's lab, and he came to work on spindle checkpoint in human cells. And he was forever going on at me about how human cells are really good, and this, why do you work on this yeast stuff? And I was forever saying this human stuff's a waste of time. Yeast are genetically tractable, it's much more powerful. I was the big advocate of the budding yeast thing. He finally, though, converted me to the dark side, right? So he finally got me interested in this idea that could we find the orthologs of these budding yeast proteins in humans? So that's what we tried to do, and we did it using a really advanced technique, which is stated here, which we sort of termed faith-based bioinformatics, which is essentially do bioinformatics and you find something, the best you can find, which is honestly not that impressive, and you stand in your lab meeting and you say, here's what I think is the human ortholog, and everyone in the lab meeting just sort of shaking their head at you and going, really? Based on, a few amino acids and some few blocks of homology? So we did that, and it proved to be successful. We had a fairly decent hit rate, and we identified a series of orthologs of these budding yeast kinetochore proteins, a number of which we're still working on now. And most of these proteins were also identified in some really beautiful biochemical work at about the same time. And so the world kind of shifted around, and we shifted from budding yeast to humans. And Patrick taught me everything I know about doing tissue culture and things like this. And he was really, really kind and spent a lot of time showing me how to do those techniques. So that at that moment, when I actually moved back to the UK to set up my own lab, I decided to work only on human cells, and I just sort of dropped the whole budding yeast thing completely, and we just became a lab working on human cells. And Sarah will recognize this, because Sarah, she joined the lab at this time. She was the first postdoc in my lab, and we were working on these proteins and trying to understand them in more detail. So this is a bit of a shortcut slide now. So we sort of started, this was at the Marie Curie Research Institute where I started my lab. This is short because it was kind of an interesting time. I was like 4 years at Marie Curie Research Institute, then it was closed down. It's another lesson. Don't worry if your institute's closed down. That's kind of okay. That can work out actually for the better sometimes. In this particular case, I think it did. We are able to move to Warwick, and with Rob and Anne, we sort of established this Centre for Mechanochemical Cell Biology at Warwick, which has been going now, it's horrible to think now, but nearly 8-9 years. It's crazy, right? Time has flown, right? But we sort of set this centre up really to sort of try to understand cell biological processes from sort of a biophysical perspective. And that's what we continue to do today. Now, that's the kinetochore today, okay, most of it. I've left some stuff out. This is a human single microtubule binding site in the kinetochore, and you can see it's an extremely complicated structure. And I've sort of highlighted some of the most important complexes and components there for you to see. If I get my pointer here, it might be useful now. So yeah, so here's some of these new proteins that we found, and you know, we're not going to go through the details of this now. The point to show you is that if you compare it back to that textbook view of the kinetochore in 1997, things have moved on considerably, and really now the task is to understand how that thing actually works. And so I've been sort of thinking this is the same logic that we used in budding yeast, which was that can we sort of say if there's separate subcomplexes, can we map specific functions to subcomplexes? That's the kind of the idea, and is that possible to do that? So that's what we've been working on. I want to show you a few examples of that kind of thing. So this is what we've been doing a lot of. This is actually filming live cell imaging of kinetochore movements in human cells. So here the kinetochores are all labelled here. This is GFP-SEMP-A. You can see this is metaphase cell. All the kinetochores are aligned nicely here, apart from this guy here. Okay, and as you'll all know, this guy here is telling this cell we don't want to go to anaphase yet, but the cell really wants to get this guy aligned onto the metaphase plate. Okay, so when I play this movie, you're going to see what happens. So we can follow this guy, and you can see him here. Watch him there. Zoom in and align himself onto the metaphase plate. It's fantastic. It works so efficiently, really well. And actually, this is a situation where everyone else, all his friends have already made it to the equator, but he makes it too. So by filming these single chromosomes aligning, we're able to sort of ask questions about what components and what factors are required for that process. And it turns out that there's sort of two main mechanisms by which that process happens. The first is that I alluded to at the beginning, which is that kinetochores can actually slide along the side of microtubules in a line. We call this lateral sliding. And the other one is Pac-Man, which you've seen before. And this is a cartoon that Phil Auckland in my lab made to illustrate to you the idea of Pac-Man as a kinetochore. So there you go. So the kinetochore is able to probably grip hold of these curved protofilaments as the microtubule depolymerizes and hold on to that, and therefore couple the sort of the energy released from the microtubule depolymerization to the movement of the whole chromosome as a whole. And those sort of those high resolution red lines are the protein components that we need to identify. And so the first experiment I want to show you is an example of the kind of things you can see. So let's just zoom in here on this complex here. This is the CCAN. One part of the CCAN is the SEMP-O subcomplex of this structure. And I want to show you here, this is just a movie of similar as I've just shown you, but here I've just depleted the SEMP-O complex, and you can pretty clearly see what happens. Lots of kinetochores align, but you get these guys here, which all effectively are stalled. They don't move. They're unaligned. They can't make movement in towards the metaphase plate. And so that sort of tells us that we think this complex is important for generating some force or getting this thing moving. That's, of course, not enough. And so one thing in the lab we want to do is also to try to look at the biochemistry of this and sort of reconstitute some aspects of this. So just on the right here now, this is an example of that, where we've now sort of reconstituted this the SEMP-O subcomplex in vitro. This is with AndrewMcAinshTrimmed. And if you take this subcomplex and label it with Alexa 488 and draw chymographs along microtubules, which you can't see here, you can clearly see here that the SEMP-O complex can bind and diffuse along the microtubule lattice. So we think that this SEMP-O complex here is able to actually interface with the microtubule, and that's an important event in doing this. And we're really... This is nice because actually depleting various components of this complex also give rise to problems in the control of microtubule dynamics. So we think that the SEMP-O complex plays some role in generating force and movement of the chromosomes. But what about the one that actually couples it to the depolymerizing microtubule? Well, it turns out that we think that's this SCAR complex here, which is I've highlighted here. Here's a zoom of it. This is sort of based on structural biology experiments, what it would actually look like. I didn't say this. I should point out all the stuff is drawn to scale here as best I can. So one tubulin heterodimer is about 8 nanometers, so the whole thing's drawn to scale to the nucleosomes and so on and so forth. So this gives you an idea of what it actually would look like. If you deplete the SCAR complex or you knock out the SCAR complex by CRISPR-Cas9 or whatever you want, you get this really cool phenotype, which you're already seeing here running in the background, which is the chromosomes actually can make progress towards the metaphase plate, as you see here, but when they get quite close, they suddenly let go and rotate away and fall away from the spindle. So this is, I guess, a sort of cartoon representation of what's happening. I'm obviously not showing you all the experiments that demonstrate this is actually what's happening, but this is what's happening. Effectively, the leading kinetochore, the one that's trying to hold on to those microtubules as they shrink, is failing and it's letting go, and that's causing the chromosome to fall away. We called that phenotype flipping, kinetochore flipping, and we can show that that's dependent on the amount of force being applied onto that kinetochore. Now, the scar complexes involved in that process made sense because actually Julie Welburn, when she was in Ian Cheeseman's lab, had actually provided evidence that the scar complex might track microtubules as they shrink in vitro, reconstituted, and actually we're able to show the same thing on dynamic microtubules. So this is now a GFP-labeled scar complex, and you can see this scar complex is, I love this movie, right? Look, it literally just tracks the shrinking microtubule. So the scar complex autonomously contracts shrinking microtubules. If you eliminate the scar complex from the kinetochore, kinetochores can now no longer load bear onto those microtubules as they pull. And in fact, very recently, there's a really nice paper from Tricia Davis's lab that's just come out where she actually tests this directly and shows that yes, indeed, the scar complex is required to form a load-bearing attachment if you apply forces on it in vitro. So it seems that this model is right, that the scar complex we think of is the load-bearing component of the kinetochore, which is why I write this here. So the idea is that we can map load-bearing device onto the scar complex. I've shown you that the CCAN thing is important for controlling microtubule dynamics in some way. And the same is true for these other complexes as well. And here I've just have simplified my complicated kinetochore by showing what I think is sort of like the core infrastructure of a kinetochore. So this is the NDC80 complex. Okay, this is the absolutely required for microtubule binding. So if you remove the NDC80 complex, kinetochores can no longer bind microtubules at all. Okay, it's absolutely required. Okay, nothing else is absolutely required. Everything else just helps. This guy is crucial. Here's the nucleosome, and the CCAN, of course, acts to link the nucleosome, the chromatin, to this outer kinetochore. There's this mysterious complex here called the RZZ complex. It was mysterious until last week when about 14 different papers seemed to come out on this complex at exactly the same time while we're working on it as well. But that's kind of good as well. That took me back to my postdoc time. The same thing seems to happen, which is, I think, a good thing. And the RZZ complex is emerging as being very important in the outer kinetochore. We think it's required for loading MAD1, MAD2 checkpoint proteins, as well as loading MAD1, MAD2 on sort of what are more their canonical platform. So we're trying to understand how these two complexes work and how those complexes relate to loading checkpoint proteins and to these other components. The other component I want to point out is this wiggly snake here. This is KNL1. KNL1 is a disordered protein. It's a very large protein. It contains multiple phosphorylation sites for the MPS1 kinase, and essentially it acts as a phosphodependent binding platform for these checkpoint complexes onto this. You'll see why I've told you that. It's going to be important in a second. So that's fine. That's what the kinetochore looks like. We can sort of start to map functions onto those structures, and that works well. But the question is, this is actually mitosis from the 19th century, and actually mitosis in the 19th century looks very similar to mitosis in the 21st century. Things haven't seemed to have changed that much, but actually, because most of what we do is we just look to whether chromosomes congress or not, or whether anaphase takes place or not, or very sort of simple things. And in fact, we need to have a much better grip on the dynamics of kinetochores and the sort of quantify how chromosome movements work. And so we made a big step towards that and thought that was a very important thing to do. And this is thanks to a great collaboration with Gauden's Dan User and Jason Swedlow and Patrick Moraldi. And I had the luck to work with those guys to develop this kinetochore tracking assay. So this is effectively a movie of SEMP-A again. And we have sort of a semi-automatic process which the software will automatically track all of the kinetochores in 3 dimensions in the human cell, and that's great because that effectively then generates trajectories. As Nigel kind of alluded to this before, you generate trajectories, and from that you can measure anything you'd like to measure about the sort of the properties of those kinetochores. And that's what we did in that first iteration of that. Over the last few years with Nigel, we've sort of advanced that. So this is the most recent version of this assay where we can now effectively in 3D track all the kinetochores and the 2 centrosomes. You saw the centrosome tracking in Susanna's talk before. This is in mitosis. You can automatically track all the things. This is now looking every 2 seconds in 3D through metaphase, and you can get like exquisite detail on the dynamics of kinetochores, and of course start to extract the properties. And all the software is open source, and you can just go and use it if you want. So this is very powerful way to ask questions of what proteins are required for in mitosis. And as Nigel just showed you before, and I've been very lucky to work with Nigel because he's brought a whole different view of kinetochores and biology and how we can try to understand that. And using these assays, as he alluded to, we can start to say something about the forces that might be acting on kinetochores and start to ask which proteins are required for generating those forces. And as he showed you, the idea from this work is at the moment is that we have this sort of tension clock. So the idea is you have two clocks on the kinetochores, which is effectively microtubule dynamics and turnover, and that there's sort of tension between the sisters, and this is important for controlling the directional movement of the chromosomes. So that's where that's kind of going to the moment, but it leaves us with a question, which is, I've shown you a very static view of the kinetochore shown here. And so the question really is, how do we know how the kinetochore itself responds to force? Okay, so can we actually ask, does the kinetochore itself change when it has forces applied to it or microtubules bind to it? How could that work? And then how would such changes be related to the checkpoint and these error correction processes? Because we need some way to connect those two things together. And so we've done some quite sort of really simple experiments in some way, which is just to use these sort of drugs to try to probe how kinetochore architecture responds to changes in force and occupancy or microtubule binding. So you can add nicotinamide, which removes all of the microtubules. That's easy. You can add Taxol in a regime where you remain microtubule bound, but you lose the tension between the two sister kinetochores. Or you can do nothing at all, and the two sister kinetochores are under tension, as read out by the distance between the two kinetochores. And so you can use that sort of a proxy for the state of those kinetochores. And we might want to ask, well, does the kinetochore organization change in those three different sort of situations? Okay. I'm going to tell you that it does change, right? So that's what I'm going to show you in a second now. I'm going to show you how we've done that. It's a bit tricky because kinetochores are very small. They're diffraction limited, essentially, at least in human cells. And what we can do is we can, of course, stick a fluorofluor, a red one, on this protein component here. And we can stick a green fluorofluor down here on this one. That's fine. So this would be single microtubule attachment. But imagine what happens. Of course, there's multiple attachment sites, so you have multiple. So you have like a line of red fluorofluors and a line of green fluorofluors. But everything's diffraction limited. It's quite small. So when you look in the fluorescent microscope, all you see is a green dot and a red dot. Okay. But what we can, of course, then do is find the centroid of the green dot and the centroid of the red dot, and we can measure the distance between those two positions. And that gives us a readout of the sort of 3D Euclidean distance between the average position of all of the molecules in these kinetochores. Okay? So that gives us sort of an idea. This was all developed by Ted Samlin's lab, and we've sort of taken the same approach and used that now to sort of probe how the kinetochore changes under these different force conditions. So I'm just going to show you some schematics. I'm not going to show you any data. I'm just going to show you the schematics of what happens because it's kind of interesting, I think. So this is the kinetochore as it stands, just normally, and this is work done by Emmanuel and by Chris. And you can see when I'm going to remove the microtubule now. Okay, it's the first thing I'm going to do. And in fact, I'm not. I forgot to tell you this. What I'm going to do is I'm going to label multiple components in the kinetochore like that. Okay, and I'm going to look at the distance between all of these different fluorofluors and this red fluorofluor. Okay, in multiple experiments, and that's going to allow me to calculate the overall organization of this kinetochore under different situations. Okay, and this is nice now. Now, of course, we're sort of building knock-in cell lines, so you can look at endogenous protein levels, and that's really useful thing to do. Otherwise, you rely on antibodies, which, as we've heard, can vary. So what happens? There's the kinetochore. Let's remove the microtubule. That's what happens. Okay, there's a huge reorganization. You can see the RZZ complex moves outward slightly. KNL1 completely unfolds. Look at that, okay? And you can also see the NDC80 complex undergoes this lever arm motion where it flips in, okay? And the MPS1 kinase is sitting right on here. That's the kinase that phosphorylates this array to load the bug proteins onto the kinetochore. So you get this big reorganization of the kinetochore. This is what happens if you just add Taxols. This is the microtubule still bound. There's no pulling forces. The KNL1 is still unloaded, but now the NDC80 is in a straight conformation and the RZZ complex has moved back in again, okay? The details of this don't really matter, okay? The point of this is to say that the kinetochore goes through these different conformational states that we can detect, okay, in these experiments. And we think it's going to be very important. This is to summarize that. Essentially, we've so far seen that there's three completely different conformational architectures of the kinetochore, when either it's unbound from microtubules, attached properly, or attached no forces, or attached and pulling forces applied. And the interesting thing is that at the same time in these experiments, we can monitor the loading of these checkpoint proteins. And as you'd expect, when there's no microtubules, you'll load tons of these checkpoint proteins onto the kinetochore. Whereas actually, in this conformation here, you actually load very little amounts of checkpoint protein on there as well. So we don't really understand exactly what's going on here, but it certainly says to us that in the kinetochore, there are a series of sort of molecular switches taking place, and these are important for coupling the behavior of the chromosome to the checkpoint and the signaling from that checkpoint. And that relates, of course, to the spring tension that connects the two kinetochores themselves and these microtubule-dependent clocks. And I haven't mentioned that we've also shown that the kinetochore itself can actually rotate freely within the microtubule bound state, and we call that swivel, and that allows the chromosomes to bind and find their correct orientation within the spindle. So we've got these multiple mechanical elements occurring within the structure, and hopefully you can see that we start to identify the proteins that are required for those things. Now, one limitation is photobleaching, because I've shown you everything in live cell imaging I've done is like only in metaphase for very short periods of time. But now actually using this lattice light sheet microscopy now, we can now film the whole of mitosis in a single movie. So each of these stacks is every 4 seconds. So we take full stacks every 4 seconds for 2 hours. Okay, there's zero photobleaching, and cells go through mitosis to completion. So now you can see here we're able to sort of render the positions of the kinetochores. And of course you can see that we're now going to be able to track all of these kinetochores through the entirety of mitosis and start to try to understand all of the processes that are taking place. And you can see here an aligned kinetochore, of course, waiting, aligning before the cell goes into anaphase. So I think this technology is going to be very important for asking a whole set of new questions about how kinetochores work during mitosis. Now, before I finish, I just want to go back to what Binyam talked about earlier, which is the process of meiosis. Okay, so because actually the meiotic divisions are really interesting. I mean, we know from what Binyam gave you a really nice introduction to this, that you have to do this initial anaphase where you segregate the homologs, so you do a reductional division, and then you do you segregate your sisters in anaphase 2. Okay, and these meiotic divisions in human cells are very poorly understood. Okay, and so we've started to look at some of this, and these are the same reasons that we've talked about before. This has massive impact onto human health, really, and human women's health. And I think it's really important to understand these mechanisms. So we've started to look at this in collaboration with the hospital and looking at human meiotic kinetochores, which is what we'd like to look at. So this is a human oocyte, and you can see here's the zona pellucida, here's the oocyte itself, and you can see here the chromosomes labelled here. This is just Dapia labelling and crest labelling of the kinetochores themselves. So you can see the thing here. And we do this in 3D, and you can go and you can look at single bivalents. Okay, so here's a single bivalent. Now what you can see hopefully really clearly here is that there are 4 kinetochores. Now the interesting thing is that in all other organisms known, these kinetochores get fused together so that you get co-orientation during meiosis 1. Okay? Even in mice, they get fused together. In human oocytes, they're not fused together. You see separated kinetochores. That creates a big problem for a cell. It has to somehow coordinate those 4 attachment sites to align those bivalents during meiosis 1. We don't understand how that works. The other really interesting thing is that the distance between these sister kinetochores in meiosis is completely maternal age dependent. There's a linear correlation. So as the eggs get older, then the distance between the sisters gets bigger, and that would presumably potentially cause more problems and increase the possibility of doing non-disjunction. So now we want to now go back to these cells now and start to film these live and to apply all the technology from mitosis to try to understand kinetochore dynamics in meiosis and understand how that process works. And I want to finish by showing you very, very new data from the lab. So what you're going to see here is this is actually mitosis 1 of a human for the first time, as far as I'm aware. So this is live cell imaging of chromosome of anaphase in a mitosis 1 division. So you can see there's 2 polar bodies here. You can see the centralization of this spindle, and you can see the chromosomes being segregated here in the first mitotic division. So that's a fertilized embryo. So I think we now have the tools now to start to probe these processes, not only in meiosis, but also in these first cleavage divisions of the human embryo. And I think that's going to be very, very exciting for the future. So that's it. I'm going to say thank you to lots of people. I'm not going to go through them all. They all know who they are. But these are the current members of the lab and all the people who have been through the lab. I haven't had time to mention many, many people who have contributed to this work. And they all know who they are, okay? And they've all been super helpful. This is where we are. This is the Centre for Mechanochemical Cell Biology. We have lots of opportunities to come and join us. If you're interested in any of the ideas that we've been talking about today here, write me an email. There's always good opportunities to come and join and take part in some of these experiments. I have to thank all the people who have funded this work. Janelia for access to their Lattice light sheet at the time, and also the hospital which is where this sort of future work and working with human embryos is going to work from. At that point, I'll say thank you very much. Oh, questions? Yeah.
Yeah, thank you very much, Ian. It's time for... sorry, not Ian. I'm thinking of Ian Hagen. Because you mentioned him. Andrew. Time for a couple of questions. Yeah.
I can just shout.
Okay. Hi.
I was just... I know you ditched them a while ago. But I was just... The budding yeast. The fact that they have a socket-like structure, does that mean that they can only do Pac-Man and not sliding? I guess kind of related.
They can do sliding, yeah. So actually, there's some really nice work on that from Tomo Tanaka's lab where, yeah, they show that the kinesins can drive lateral sliding of those kinetochores too. It may be there's some reorganization of that kinetochore when it goes from being side on to end on, and that conversion from lateral to end on attachment actually quite poorly understood. How does that happen? That's not really known. That's a bit of an open question. So yes, I didn't ditch budding yeast. They're still great.
So I wanted to, out of curiosity, what happens with all these proteins when the cell is not dividing? So are all these complexes stored just waiting there, or do they do something else?
Yeah, I mean, there's a huge assembly of I didn't talk about that. There's a huge assembly of the kinetochore as it goes through G2 and goes into mitosis. So that CCAN, that pink thing, complexes that I showed you, that's the constitutive complex. So that thing sits on the centromere all through the cell cycle, just sitting there. Okay, it's there all the time. The rest of it just gets loaded onto there as you go into mitosis, all probably driven by CDK cyclin activity as you go into mitosis. This whole thing assembles. Before then, I think it's just floating around. Yeah, I guess so, right? I don't know. I assume it's just floating around.
Hi, hello. When you live image kinetochores, can you tell on which particular chromosome each dots are on, and can you see chromosome-specific behavior of kinetochore? Because I'm asking because Jan and Enberg published some kinetochore always segregate earlier than others, and that determines the position of chromosomes in interphase.
No, that's a great question. So I'm actually not working on that. Sarah is working on that. So you should ask Sarah at Coffee all about that.
Okay.
The answer to your question is yes, there are biases. So yes. So there are certain chromosomes that have predisposition to being missegregated. So I think there are biases. I think the molecular basis of that, I think, I'm looking at Sarah now, she's going to hopefully nod at me. It's not that well understood. She's going to figure that out. I think it's a really interesting question. One issue that we have, what we'd like to be able to do, of course, is label in live cells each of the chromosomes using, say, for example, CRISPR-Cas9 or something like that, so we could potentially label them. So that's something that would be interesting to do as well, for sure. It's a great question.
Okay, yeah. I'll visit her later.
You should visit her later.
Yeah, thank you very much.
So, with the sort of microscopic hat on, what happens in your... Have you kind of looked at your movers as the kinetochores disassembling at the end? So basically, there's a direction of travel in the phase, you get to the end there. Is it that it just all falls to bits, or is there an order?
Yeah, no, that's a good question. So I have to say that for a long time I was quite worried about myself because I found that I would have been working on metaphase is 10 minutes long, right? So you've got 24-hour cell cycle, and I'm like working for years and years on 10 minutes, right? There's one phase because it's the most tractable phase to look at. I've never looked that late. I mean, we're starting to look at anaphase now, that's how desperate it is, right? So now we start to look at anaphase and understand what happens there. That's not really very poorly understood. That's poorly understood in its own right. And actually, later on, I don't know how that disassembles. I think that's a really interesting question. We've never looked.
Okay, thank you very much, Andrew, for a fascinating talk.
No problem. Thank you, everybody.